Un minuto, Ramón.
Ya se está transmitiendo, doctor, adelante.
Okay. Welcome, everybody, to this very interesting seminar. We will continue with our celebration of 20 years from the conversion of Center to Institute, Neurobiology Institute. And for today, our speaker is, I'm very, very glad to introduce, is Professor John Dani. I'm going to talk a few moments about his academic biography. He got his PhD at University of Minnesota. He did 2 postdoctoral. The first one in University of Washington in the field of physiology and biophysics. The second one at UCLA in physiology. Eventually, he arrived at Baylor College in Houston, Texas. In that place, he was promoted after several years, and eventually he became director of the Center on Addiction, Learning, and Memory. Some years ago, he was incorporated to the University of Pennsylvania, again as a director, but in this time of the Mahoney Institute for Neurosciences. Professor Dani is an expert and authority in the neurobiology of addiction. He has done very, very interesting contributions studying cellular mechanisms about how nicotine, cocaine, and other drugs can affect the function of the brain. In PubMed, you can find more than 130 publications of John Dani, many of them in top journals, such as Nature, Nature Neuroscience, Annual Reviews, Molecular Psychiatry, and others. He has done excellent contributions in the neurobiology of addictions, studying in the last years the nicotine as an example of drug of use. And in this time, he will talk about nicotine, but in addition with stress and what happened with the ethanol auto-administration. Thank you very much, John. Be welcome, please.
Thank you very much, Dr. Diaz. It's flattering to have been invited. I appreciate the opportunity. And let's see now if we can get everything going here. Okay. Nothing's gone wrong yet. What a miracle. Here we go. There we go. So again, thank you very much for inviting me. I'm quite flattered that you're interested in hearing about the work from my lab. Some of the people that all of these are members of my lab who did work that I'm about to show you, particularly Blake, Alexia, and Elise, and I'll talk more about them at the end of the talk. I'm going to begin by focusing on a particular pathway where we talk about the idea of associative memories, that is, memories that are associated with the act of using an addictive drug. And I'm going to focus on the perforant path, which takes processed information about the environment and brings it in, and this is a very oversimplified picture of the hippocampus. And one of the places where the perforant path makes its first synapse is onto the granule cells of the dentate gyrus. And there, long-term synaptic potentiation, we know after 50 years of work, is one of the mechanisms for memory. And I'm going to focus on what are the system level, the big area picture of what nicotine does when it arrives in the brain, and particularly when it arrives in the hippocampus. And our point of view is the following. The most serious problem facing the field of addiction is relapse after years of abstinence. For instance, if a person didn't relapse, then addictive drugs wouldn't be a problem. You could experiment with addictive drugs, and then when you're 22 and have to grow up and have to go to work, you could stop using the drugs. But the problem is some people relapse. And one of the important reasons why they do relapse is the environmental cues where they use the drugs and the situations under which they use the drugs. When they see those and experience those environmental cues, it elicits drug-associated memories, which can be below the level of consciousness, such that it produces internal states. And I'll talk about that in a moment. And those internal states cause motivations or cravings that will eventually can lead to relapse. And the way we did the experiment to begin with was in freely moving mice. We did an experiment much like you might be able to do in a slice, but we did it inside of the animal while it was freely moving. We put a stimulating electrode down into the perforant path, and you can see this is an extremely big pathway carrying a great deal of information, particularly about the context and the place in which the memories are occurring. And while we could stimulate the perforant path, we recorded in the dentate gyrus of the hippocampus. And this would be a typical recording where this is the stimulus artifact. This is the fiber volley, the stimulus coming in on the fibers. This is the postsynaptic response in the granule cells. And this is the granule cells firing action potentials as a consequence. If you stimulate this pathway to undergo long-term synaptic potentiation, LTP, which is a substrate for memory, then this stimulation would have a different consequence. As you see, the input is the same, but the downstream consequence is much larger because of the synaptic potentiation. So this is a very precise measurement, and it's done in real time from a mouse, where we just use a, you can inappropriately call this a gain of one amplifier. It's actually a FET on the head of the mouse. And this was a picture we took literally one of the first times we ever made this measurement about a decade ago. And these have all gotten much smaller and nicer now. But the mouse is not learning anything. By the time we do our experiments, it has been brought up into a soundproof room and then put into a soundproof box. And inside that box, it's in a tube. And that when that box is closed and soundproof, it's completely dark. And this has happened many, many times. So the mouse isn't learning or undergoing synaptic plasticity because of the situation. It's only what we manipulate as experimenters. And what we did was the following. We measured the synaptic transmission the day before, and we required it to be stable. And then the next day we would come in and inject, let's say in this example, saline. And then we would follow it for 3 hours, sometimes 5 hours on that day. Then the next day it had to be stable. The next day it had to be stable. And 5 days later it had to be stable. And we would go back with this stable recording and inject a relatively low level of nicotine. And we injected this relatively low level of nicotine, you see nothing really happened. Remember, this animal's in a dark room, it's silent, we don't reach in to do the injection. The injection's done through a tube that we've implanted in the animal. So the only stimulus it has is the nicotine or the saline coming into its body. But then we injected a high dose of nicotine, 10 times higher. And in that case, you induced long-term synaptic potentiation measured in real time. This is the first time this measurement was made where an addictive drug in real time inside of an animal, the only stimulus, induces long-term synaptic potentiation of the kind that underlies memory. And the recording was stable for days. And if we blocked nicotinic receptors before we did this, that would prevent this LTP. So it did depend on nicotinic receptors in the brain. Then we wanted to explore what was going on in the inhibitory circuitry around the granule cells. So when we stimulated the perforant path, it would excite the granule cells and they'd shoot those action potentials if the stimulus was strong enough. But at the same time, some of the stimulation would go to the surrounding inhibitory neurons, and a moment later, that would arrive at the granule cells. So if we set in different patterns, different frequencies of stimulation, the inhibition and excitation would collide, and we could measure how strong the inhibition was. And when we did those experiments, we found that both this feed-forward inhibition and the feedback inhibition, both of them were decreased inside of the animal's brain, in the hippocampus, before we would see the induction of LTP. So the nicotine arriving somehow decreased the local inhibition, which enabled LTP to happen. That was the assumption we made based on these in vivo findings. So what we did is we began cutting slices and looking at this area in slices where we could have more experimental control. And we patch clamped a granule cell, and we can still stimulate this perforant path. And what we did is we went into the hilus of the dentate gyrus, and this is the granule cell layer where the granule cells are located. And we did single cell PCR in the many different inhibitory neurons that were present in this area. And we focused finally, through a lot of work, onto these hilar interneurons that project into the perforant path. So these are the hilar neurons. They're right next to the granule cells. And they have very dense processes up in this area where the perforant path comes in. And this is didactically represented in a cartoon over here on the right, where these are the granule cells, and here are the hip interneurons broadcasting inhibition, ongoing inhibition, up into the perforant path. And this inhibition is very potent. So if you were to put a recording electrode into the granule cell layers in a freely moving rat or mouse, and just let it sit there, it would mostly, if it's in the granule cell layer, it's mostly silent, because there's intense inhibition in this area. And only occasionally would you see a couple firings or a little of firing, then it would go silent again. And I know this because many graduate students have gone into this area to record and spent a long time getting not much. So here you have this ongoing inhibition coming in. But when nicotine is applied to the bath at about the same dose that we gave it in vivo, what we found is other GABA inhibitory neurons would inhibit the hip neuron, come in, and they'd inhibit the hip neuron, so this ongoing inhibition could no longer get through. The hip neuron is inhibited, and now this area is disinhibited. So it's free to read this perforant path input that was previously blocked. So you're saying, if you can do this in a slice, why bother doing it in vivo? To get an N of one measurement, if everything goes right, takes 1 month. We have to measure the saline, low nicotine, high nicotine. It's a week for each of those measurements to do it properly and get in and out, input-output curves to make sure your recording is stable. It takes a month. So why not do this in slice? It's so much easier. That's because this level of nicotine in slice, even though you can study all these synaptic mechanisms much more easily, they do not produce long-term potentiation of this synapse. So there's something else that's going on. And we had done work previously and shown that when nicotine arrives in an animal, it evokes a dopamine signal. And we reasoned that that's what was happening. And in work that I'm not going to show you, we have shown in slices that dopamine enables long-term potentiation to happen. It makes it easier. Normally, synaptic potentiation requires presynaptic activity and a postsynaptic response. And that presynaptic and postsynaptic activity has to be very precisely timed, and it has to be sufficiently strong. So only when it's precisely timed and sufficiently strong are you able to get LTP. But if there's dopamine bathing the area, it lowers the threshold. The timing doesn't have to be as perfect, and the strength doesn't have to be as hard, and it's much easier to get LTP. In fact, you may not get it under the same circumstances without the dopamine. So we went back in vivo and we inhibited D1 receptors either everywhere in the brain or locally down here in the hippocampus. So we injected saline, then we injected that level of nicotine that produces LTP, and we measured it in vivo. Then later, the next week, we came back and we inhibited the D1 receptors locally in the hippocampus, and then that level of nicotine no longer produced LTP. That is, if we inhibit the D1 receptors, the dopamine response in the hippocampus is blocked, and then LTP is prevented. So then we can ask, does the addictive drug nicotine enhance meaningful LTP induction along the perforant path? Remember, this mouse has been coming up into this box over and over and over again. It's not really learning on the environment or anything anymore. That's long past learned. But now the nicotine itself seems to have been able to induce LTP. Well, what if there is information along the perforant path, new valuable information? We mimicked it by weakly stimulating with theta burst activity along the stimulating electrode. Theta activity is a waveform that you'll see in the hippocampus of rodents when they perform, let's say, run a maze or perform a task. While they're running a maze, there'll be waves of theta activity, waves of it passing through, moving through the hippocampus. And this theta activity enables learning and memory to go on. So we mimic that by introducing weak theta activity in the stimulus pathway and recorded in the hippocampus while we conducted our experiment. And again, we injected saline in this weak theta burst activity. We didn't see anything, and we had a stable recording. But if you remember that low level of nicotine that by itself didn't do anything, 0.1, well, this time when we injected that low level of nicotine while introducing weak theta along the perforant path, we induced LTP, and it was so potent, it was statistically significant even the next day. So you can reason or hypothesize, and I think the better word is speculate, that what you're seeing here is that the pathway, the perforant path, carrying information for memories is enhanced in association with the drug experience. That is, you're getting associative memory. That is, while I'm taking the drug, whatever place I'm in, whatever people I'm with, circumstances, that information coming in on the perforant path, which carries information about place and context, is undergoing the plasticity of the kind that is learning and memory. If we block the dopamine signal coming up into the hippocampus and repeat this, the plasticity goes away, the LTP goes away. So you can again speculate that the association between the addictive drug and the memory is prevented when the dopamine signal is inhibited. And let me take a moment, an aside, for those of you who are experts out there are saying, you know, looks like you block the dopamine signal from the midbrain, but a lot of the dopamine signal also comes from the locus ceruleus. Well, this would be a separate talk, and I'm not going to discuss it. I don't even have all the information with me, but we showed that indeed there is a dopamine signal coming from the midbrain. We retrogradely labeled dopamine terminals in the hippocampus and followed them back into the substantia nigra ventral tegmental area. And here they are. And one of the surprises, and the reason why I think many other labs missed it, it was expected to be out here. But when we actually did it, it's mainly in the substantia nigra and just on the border of the ventral tegmental area. And when we used two other completely independent methods to do this, they all gave the same answer. Another independent method found the same set of neurons, again, largely in the substantia nigra, and these are dopamine projections to the hippocampus. So indeed, not only the locus ceruleus, but also the midbrain dopamine centers of the ventral tegmental area and substantia nigra project potently up into the hippocampus. So let me summarize what I've talked about so far. When a person uses nicotine, nicotine goes everywhere in the body and the brain. And when it arrives up in the hippocampus, it causes local action such that it disinhibits the granule cells. So only during this period of time are they free to really feel the impact of the perforant path, and it's potently being transmitted and accepted and driving the granule cells. Well, this is at the same time, the information it's going to be sending is, you're smoking cigarettes, you're out in the parking lot with your friends smoking a cigarette. That's the place, that's the context, and that's the memory that's going to be put down and associated with the drug. But the memory and the synaptic plasticity for the memory can only happen because of the long distance action of the nicotine on the dopamine centers, including the locus ceruleus, but also the midbrain, broadcasting a dopamine signal up into the hippocampus, which lowers the threshold. That is, not only are you driving the granule cells, you're producing the synaptic change more easily because the dopamine was there. If it weren't, you would get the information to the granule cells, but you aren't going to be likely to remember it over long term, which is the reason you can get relapse. So what about real behaviors? What I've talked about so far, pretty much experimenter controlled. But if we look at a rewarding behavior, such as conditioned place preference, in which an animal is put in a box that looks different and given nicotine only on one side, the animal will learn if that nicotine dose is precisely correct. You have to get it exactly right. The animal will prefer that side and stay on that side 100 seconds compared to 10 seconds on the other side. But if you inject with saline, the animal will still prefer that side, but if you block the D1 receptors, it will no longer remember to like that side. It will behave as if nothing has happened on either side. What about an aversive memory? In this case, a mouse is put into a lighted box and there's a light on and they don't like to be in the light, and a door will pop open and there's a dark chamber here that's in the dark, and the animal will quickly go into that side where it's preferred. We close the door, let the animal in there for a moment, and then give them a shock and leave them in there for 2 minutes to learn this space. If we block D1 receptors, either in the hippocampus or everywhere, and repeat this experiment, as the shock gets stronger, the animal will wait longer, in this case 100 seconds, before it goes into the other side. But if the D1 receptors are blocked, it doesn't wait as long. As a matter of fact, until the shock gets very strong, it doesn't remember to avoid the place at all. And when we did local infusions into the hippocampus, we got the same result. It seems like D1 receptors in the hippocampus are important for learning this event. And as a matter of fact, if when the animal did the experiment, if we gave an injection to excite D1 receptors with SKF, we gave an extra excitation, not only the normal dopamine signal, but also this drug that enhances D1 activity. If we look 72 hours later, the animal remembered better to avoid that place. So not only is the dopamine signal sent by the addictive drug helping the animal to remember by working through these D1 receptors, these data suggest that the stronger the dopamine signal is that you send, it's like a scalar. It scales up how good the memory retention is going to be. So if I use a lot of the drug and sending more of a dopamine signal, I'm going to remember the context place better than if I hadn't. So the suggestion here is that the in vivo synaptic plasticity does not occur if these D1 receptors are inhibited during rewarding or aversive inhibitory avoidance behavior. And if you block the D1 receptors, you don't have a hippocampal dopamine response, you don't get the memory and plasticity. So what we've shown here is that the environmental cues that elicit drug-associated memories or these internal states can be the motivation for relapse. And this is one of the pathways. This is how it works. Normally, that pathway is helping you remember how to conduct a good life. How do I find the places in the context where I had a successful behavior? But the drugs of addiction, in this case, nicotine, is usurping these normal mechanisms and creating memories that when they come up below your level of consciousness, they induce an internal state that makes you want to seek the drug. Let me give you an example. If you haven't had anything to drink, or you've gone out on a hike out in the desert and you run out of water, you get really thirsty and you feel it everywhere. You just desire the drink of water. And when you get to the water, you're just driven to get that water. You drink it, and after you've drunk enough, you no longer feel that. The internal state that drove you is gone. It just went away. And I've never been addicted, but I can imagine these are comparable kinds of feelings when I've talked to people who have been addicted, when they describe what the motivation is like. And this is happening below your level of consciousness using the same mechanisms, the same evolutionarily very old systems that normally shape your behaviors for success, and they get usurped by the addictive drug. Now I'm going to talk about the link between nicotine and tobacco and alcohol. There's a lot of epidemiology. In fact, the epidemiology is what spurred us to go do this work. But here I'll give you just a taste. People who are dependent on tobacco are 4 times more likely to be dependent on alcohol. And the link is so strong that alcohol and tobacco is so significant that the statistics indicate that more alcoholics die of tobacco-related illness than die of alcohol-related problems. So it's a very strong link. And you may remember back in the days when you were going to bars and smoking and drinking in bars, very often bars are just filled with smoke because people who were drinking were also smoking. And so that drove us to think, this is so strong. The epidemiology is overwhelming. Something must be going on here. It's not just, well, people who like to drink like to smoke. This is probably real. So we started with a very simple experiment. We started doing microdialysis down in the nucleus accumbens, which is a main center that receives the dopamine signal induced by addictive drugs. And what we did is the simplest experiment we could think of when we started. we gave nicotine either 3 hours, 15 hours, or 48 hours before we did microdialysis to an ethanol injection. And I'm using those 3 time points because they all gave the same result. What we found was when we injected saline and measured the microdialysis, we got a nice dopamine signal induced by ethanol. But when we looked 3 or 15 hours or 48 hours after we gave the nicotine dose, the microdialysis found a smaller dopamine signal. And we didn't expect that at first. So we've made this measurement, I don't think I'm exaggerating to say 100 times. Here we probably have an N of, on this slide, maybe an N of 10 or something, but we've done this many, many, many times over the years now. And these are solid data. Associated with this same time point, if we start operant alcohol self-administration where a rat pushes a lever to get the alcohol, we found in that same time frame, the animal that's experienced the nicotine will administer statistically more alcohol. So there's some kind of association. And we also found at the same time that when you give an animal nicotine, there's a boost in their stress hormones. Corticosterone is boosted during the same period of time. And there was work in the literature that led us to hypothesize that the stress hormone signal initiated by nicotine could cause subsequent alcohol consumption. The link was already made in the literature. And so when we found this, we rapidly went to this hypothesis. So we decided, let's inhibit the glucocorticoid receptors and reassess the influence of alcohol over ethanol's dopamine signal. So we systemically blocked glucocorticoid receptors, gave the nicotine, and looked at self-administration of ethanol. And instead of being boosted like it normally is by nicotine, they self-administered just like the control animals, as if they were injected with salt out here, salt water. So then we hypothesized that stress hormone signals initiated by nicotine could alter subsequent alcohol consumption. And it was based on a large literature that said that stress hormones can influence alcohol drinking. And this is some of them. So we decided, let's look directly at stress. And what we did is we put Long Evans rats, and this is a Long Evans rat, there's its head, there's its body, and there's its tail in restraint stress. So it's put in this restraint stress for 1 hour. And then the next day, 15 hours later, we begin alcohol self-administration, the initiation of alcohol drinking. And it's important that you don't do this stress too close to when you do the behaviors, because when an animal has been stressed for a while, it will just hover in a corner, won't do anything. So after this stress, and you have to be sure that the animal's stressed, sometimes these rats become so friendly with you, they're so used to you, they completely trust you, and you can't stress them in this way. So you have to see that the animal defecated and urinated and its fur is all ruffled and it's unhappy. And then when you put it in its home cage overnight and come back, it will self-administer. And the animals that were stressed in this way will self-administer more alcohol than the ones that have not experienced the stress. And if we block the glucocorticoid receptors before we give the stress, the animal acts as though it wasn't stressed, and it self-administers like the controls. And in fact, if we block the glucocorticoid receptors just in the midbrain dopamine centers, the ventral tegmental area and the substantia nigra, that animal will also administer just like it didn't experience this. So you'll get comparable results when you infuse just into the DTA. So that led us to something else. At this period of time, about when we were doing this work, I was at a meeting, an international meeting, and one of the speakers was from the country Norway, which apparently they're concerned about too much drinking. And he presented a talk about epidemiology that said that the biggest predictor of a person being picked up for drunk driving or driving under the influence of alcohol as an adult was whether or not they smoked cigarettes as an adolescent. And again, I didn't believe this is just coincidence, that something's going on here. And adolescents are starting to use a lot of nicotine. Beginning in about 2015, nicotine vaping in the United States with e-cigarettes increased 9-fold in high school students and 6-fold in middle students, because they started vaping these bubblegum flavored and sugar flavored strawberry, watermelon, bubblegum. So in a sense, you were bringing in a whole new cadre of students who would not normally use any addictive drug, but this sweet bubblegum that seemed harmless, and kids who wouldn't normally smoke or use any addictive drug were starting to use it. And unlike for adults, e-cigarettes in adolescents do not facilitate quitting. Rather, it encourages the conventional use of cigarettes because they both become addicted to nicotine and they start seeing them as being harmless. So the evidence continues to accumulate that adolescents who use nicotine are associated with a greater likelihood of using not only other cigarettes, but other addictive drugs, including alcohol. And that's epidemiology spurred us to go and continue this work looking at adolescents. And what we did is we began injecting nicotine, but we also did it another way. We put nicotine into the drinking water, and both ways work the same way, in adolescent rats. And I should say, I'm going to show you the data from rats. We did the same thing with mice, and we got the same results. So in some cases, I'm even later on going to show you results where we only were working with mice because we wanted to take advantage of genetic tools. And we did it during this period from day 28 to day 42. This is a period of adolescence. Many people define adolescence as beginning earlier, and some people say it goes longer, but all the studies agree that this period is during the heart of what's defined as a rodent's adolescence for rats. And so during this period, the animal was either injected with nicotine or drank nicotine in the drinking water. Then it was put in its home cage, no more experience for nicotine for a month. And then as a young adult, we began self-administration of ethanol. If you remember what I showed you earlier, when we injected nicotine into an animal and looked the next day, they drank more ethanol. But that only lasted for 4 or 5, up to 7 days. But after 7 days, those animals became normal if they were adult. This is a month later. And what we found was that the animals that had had adolescent nicotine, when they started drinking, they drank more. And for the 7 days that we followed it, they drank substantially, highly significantly more ethanol than the controls. If we did this same experiment with young adult animals, P60 to 74, and waited 1 month, remember, that's way longer than before. I was showing you up to 48 hours or 15 hours after they experience nicotine. This is a month later, there's no effect. They drink just like controls, the adults. So there's something special, and that's not surprising to us, about addictions for adolescents. We then cut slices from this area and we started looking at midbrain ventral tegmental area GABA neurons. And we looked at the reversal potential for chloride when you looked at GABA inputs onto GABA neurons of the midbrain. And if the slice was cut from an animal that experienced saline during adolescence, it was different from an animal that experienced nicotine during adolescence. Normally, when GABA inputs come onto this GABA neuron, the resting potential begins at about -80, and the animal is pulled, the neuron is pulled to hyperpolarize and turn off near -90. But after the animals experience nicotine during adolescence, The VTA GABA neurons from those slices, when the GABA input comes, it actually has a reversal potential indicating that GABA depolarizes the cell from about -80 towards -60. That is, the reversal potential for GABA, these chloride currents, is depolarized because the chloride gradient is inside, the chloride gradient is decreased with elevated intracellular chloride. That's what this result indicates to a biophysicist. When we treated the animal with nicotine but blocked the glucocorticoid receptors during adolescence, those slices behave just like the animal experienced saline, like the controls. And likewise, if we cut slices from animals that were treated during adulthood, they also behaved as if they did not experience adolescent nicotine. The effects weren't there. What we discovered was that a pump called KCC2, which sets the chloride gradient for GABA neurons of the midbrain, but not for the dopamine neurons. It's not located in dopamine neurons. This pump, this transporter, was diminished in the slices we cut from animals that received adolescent nicotine. That is, the effectiveness of the pump was decreased, and the long-term expression of the pump was also decreased. Without adolescent nicotine, the activity would be up here. So what happens is the following. If you stimulate GABA inputs onto the VTA GABA neurons cut from a normal slice, this is upward going current, but that means chloride's going into the cell. If you pulse at this reasonable frequency, the currents stay about the same in a normal slice with inward going chloride. But in a slice cut from an animal that experienced adolescent nicotine, as the chloride comes in, it accumulates and it makes it harder and harder to drive the chloride in, so the currents get smaller. And you can quantify it based on the slopes of the evoked inhibitory postsynaptic current amplitude versus the number of stimulations. And the situation is shown here. Normally the chloride comes into the cell and it's pumped out. But when this pump isn't working right after experiencing adolescent nicotine, the chloride accumulates, isn't pumped out fast enough, and it's harder and harder to get the chloride to come in. These currents get smaller. So what we found, consequently, is that the GABA signal from the ventral tegmental area onto the dopamine neurons is altered. So again, adolescent nicotine, and then we do electrophysiology in early adulthood. And what we found is the baseline GABAergic activity in these two slices cut from these different situations is about the same. But when we bath apply ethanol at the high level a drinker would experience, we see much more GABAergic activity in the slices that were cut from an adolescent that experienced nicotine. You can see it's highly significant. These slices look normal. If you just don't look too hard, there's nothing different about them. But if you add adolescent nicotine, if you add ethanol to the adolescent slice, you get much more GABAergic inhibition onto the dopamine neurons. So we add nicotine during adolescence, we look at the inhibitory recordings on the dopamine neurons, and you see they're much elevated. If we block the glucocorticoid receptors during this nicotine treatment in adolescence, then the slices behave like the controls. And we found something even more interesting. If we bathe the hippocampal slices in 1 micromolar corticosterone, that is the stress hormone, and usually bathe it for about an hour before we start the experiment, it behaves as though it was cut from an adolescent nicotine slice. suggesting that corticosteroids are necessary and sufficient for this adolescent nicotine stress-induced change that we're seeing in the GABAergic circuitry of the midbrain, which underlies increased drinking. So let's focus on this GABAergic neurons. What normally happens is when GABA inputs onto this GABA neuron happens, you get a chloride input, but it's pumped out. It's potent. This thing is very potent, and it clears the chloride, and it drives the cell to hyperpolarize and to turn off. But after adolescent nicotine or adolescent stress, as a matter of fact, too, if it's severe, when the chloride comes in, it begins to accumulate because this pump is not only not working as well, it also isn't expressed as highly, and it can't clear the chloride. Chloride begins to accumulate, and instead of pulling the cell towards -90 and turning it off, it actually depolarizes its some. So that this is actually real data. If we look at a GABA neuron, this GABA neuron firing along, and then we stimulate GABA inputs in the surrounding area, and then as soon as we turn the stimulus off, we look at this cell, it's been inhibited. That's what we normally expect in a control slice. But in these slices with adolescent nicotine or adolescent stress, as it turns out, you get a different situation. The neuron's firing along like usual, but when we stimulate the local GABA inputs, when we take the stimulus off, the cell actually fires even faster, as if it's been stimulated. So this is paradoxical. So these are the data I showed you, but now let's use this compound that we received from some friends in Canada, the Chronic lab, which enhances KCC2 activity. And when we do that, this normal rundown that we see, shown by this dotted line, when we treat with CLP290 in a slice, it goes back to normal. And if we look at the enhanced ethanol-induced GABA activity onto the dopamine neurons, which is way up here, if we treat with CLP290, that also goes back to the control case. And in fact, if we inject CLP290 into the animal, or if we infuse it locally into the ventral tegmental area of an animal that's experienced nicotine before the day before each, it's given every other day before self-administration. The self-administration, instead of being elevated, goes down to control levels. So by boosting KCC2, we get rid of all these synaptic and cellular effects we were observing, but we also get drinking back down to the control level instead of the elevated level. And that can go on either systemic, or if we locally infuse it into the ventral tegmental area, substantia nigra area, it's kind of swimming around in that area. So if the chloride transporter, it's actually a potassium chloride co-transporter, if this chloride transporter is corrected and excitatory GABA is prevented, then stress nicotine do not produce the GABAergic circuitry changes or increased alcohol self-administration that we saw. So that led us to another hypothesis based on the literature. It was in the literature that 5-HT2A receptors can normalize, can activate depressed KCC2 activity in GABA neurons, and we just assumed it's going to work the same way in the ventral tegmental area GABA neurons. That was our hypothesis. The chloride extrusion enhancer, CLP290, is untested in humans, has never been used in humans, and probably never will be. But serine 940 is a common site of post-translational modification of KCC2. That was already in the literature. We know that if that becomes phosphorylated, KCC2 is more efficient. And it was in the literature that the metabotropic serotonin 2A receptor can upregulate KCC2 activity via kinase activity. So we hypothesized it was going to be at serine 940. So you activate these 5-HT2A receptors, they're going to activate KCC2. So it's also in the literature, this is known since the 60s, that psychedelics like mushrooms are 5-HT2A receptor agonists, and that they clinically are efficacious for treating alcohol dependence. Yet the neuronal basis for this therapeutic success has remained obscure. So we hypothesized this could be one of the features that's going on here. So we looked at this GABA input onto the VTA GABA neurons. Normally, stress or nicotine will lead to elevated chloride, high chloride, and you get this, it moves towards, this isn't always excitatory, but it moves towards excitatory GABA activity onto VTA GABA neurons. We used a specific 5-HT2A agonist TCB-2 that can be purchased. I don't know if it was Sigma or where we purchased it, but when it's given to these into these slices, it corrects this by activating KCC2. You keep low chloride, and now you go back to this inhibitory form of GABA activity onto this GABA neuron. And so normally, from a nicotine-treated slice, you get this depolarized reversal potential, but if you treat the slice with TCB, this 5-HT2A agonist, you return back to the control condition where GABA is hyperpolarizing on GABA neurons. And then what we did, we obtained a mouse from the Moss lab that has this serine 940 removed. It's replaced by an alanine. So there's no longer, you don't have that site. And when you do that, you find that all the midbrain GABA neurons, whether you treat with TCB or not, it doesn't matter. They all act as though the GABA input is depolarizing. And after you treat with stress or with nicotine, nothing really changes. It's still up here. So this kind of, along with other data, confirms that serine S940 on KCC2 is a phosphorylation site that regulates KCC2 activity. We found that when we inhibited protein kinase C internally, then the 5-HT2A agonist was ineffective. When we used inhibitors to block the 5-HT2A receptor, then this agonist also was ineffective. When the serine site was removed from the animal at that KCC2, then the VTA reversal potential for the GABA neurons were depolarized and could not be further influenced by this serotonergic agonist. And in fact, the serine 940A mutation produced the cellular changes that looked much like the treatment with nicotine or stress, suggesting that's their main site of action. So then we went on to explore this further in mice. So everything I've showed you up until now is done in rats, but we repeated those experiments in mice. And now these experiments were conducted in mice in which the mice were allowed a 2-bottle choice. They could either choose between drinking from a water bottle or an ethanol bottle. And this was every other day. If you let them drink every day, they'll drink too much, they'll be hungover, and the next day they won't drink. So they only drink every other day. Or sometimes even you can even go longer. And over time, you ramp up the amount of ethanol in the drinking water up to 20%. And over the course of weeks, they'll start to self-administer at a relatively stable rate after 4 weeks. And then if we injected those animals with this 5-HT2A agonist the day before the drinking session, those animals drink less. And it's found that they consume less ethanol and they showed less of a preference for drinking ethanol based on this 5-HT2A agonist, which we showed boosts KCC2 activity. So it has an impact on the drinking. But it does not impact the animal's drinking of water or saccharin. If we gave it 2-bottle choice to drink saccharin or water, and we gave it with or without this 5-HT2A agonist, the drinking of water and saccharin did not change. So it's not just goofing up everything. Some things are unchanged, but the drinking is decreased. So I have a couple minutes left. So let me give you the simplest cartoon summary of what was going on. If we take a normal slice cut from a control animal and bath apply ethanol, the ethanol in this simplest of simple cartoons of the midbrain dopamine centers, the ethanol will cause a glutamate excitatory signal onto the dopamine neurons, and it will cause an inhibitory GABA input onto the dopamine neurons. And that will lead to the dopamine signal that we measured in the first slide by microdialysis. But there's also, let's add one more ingredient to this circuit. And everything that I showed you, all the complex experiments I showed you about GABA, we did that with glutamate too, but what we found is adolescent nicotine and stress did not change the glutamate input to ethanol. It stayed the same. Ethanol still caused a glutamate input, but it was the same. So I didn't show it to you. But after adolescent nicotine or stress, the circuitry changes a little bit. When you bath apply the ethanol, it turns out that this circuitry now has a depressed efficacy of KCC2 in these midbrain GABA neurons, so that this input is still the same. It's still the same GABA being released, but when it hits this cell, instead of inhibiting it, it doesn't inhibit it as well and can even excite it so that more GABA is released. This is about the same, so that the input out here is smaller. And I think this is vital. There's still a dopamine signal, but it's just smaller. So you might take the most simple-minded argument. The animal takes more ethanol to try to get this dopamine signal back up. That isn't an adequate explanation. I'm sure it's much more complicated than that. But if this dopamine signal were completely blocked, I think you'd get a different result. And other types of experiments have been done when people completely disrupt the dopamine signal and animals won't self-administer. So I think this is the simplest circuitry and summary of what we just looked at here today. And I'm almost out of time, but let me just summarize by saying, when nicotine arrives, it bathes the whole brain, the whole body, and there are both long-range, local circuitry things that are going on, probably in other areas that we didn't study as well, and then these longer-range things, such that we think we've struck on something kind of important here. It's gotten a lot of publicity, a lot of recognition, and we're looking forward to doing more. And I quickly, in the little minute I have left, want to acknowledge the people. There are a lot of people in the lab who did work, but this was mainly, and Theo did the hippocampal dopamine signaling label, and that's a separate talk that I didn't give today. But two great graduate students, Blake and Elise, did their thesis work on this in this area, and they're outstandingly good. They've gone on, they're doing postdocs, highly successful, high-profile publications in the postdocs. They're doing great. And Alexei Ostromov, a Russian by way of Trieste, Italy, where he trained in biophysics in Trieste, which is an outstanding place for biophysics training, came to the lab and was really instrumental. They all were important, but Alexei really was a driver in the lab that got this done. He's now an assistant professor down in Georgetown, and he's doing just great too. Not surprised, he's doing just great. We have many collaborators, but on this project, Mariella De Biasi, who's here at Penn, a neuroscientist here at Penn, Heath Smith, who's also here at Penn, and Jinrong Tong, who was in my lab for a short time before he took his position at Baylor College of Medicine and Texas Children's Hospital, our long-term collaborators. And at this point, I will stop sharing my screen. There we go. Thank you very much.
Thank you, John. It was an amazing seminar. Remember that you can write your questions, or you can ask to your microphone be open if you want to talk.
Well, I'm happy to take any questions that anybody might have. Very,
very good. We have one. The first question is from Benjamin Villasana, his question, may the glia be implicated in the effects of nicotine on the potentiation of granule cells activity?
You know what? I missed the very first part of that because I can't see your lips. I'm having a hard time. Can you say that again?
To repeat, may the glia be implicated in the effects of nicotine on the potentiation of granule cells activity?
You know, that's an easy one. The answer is yes. You know, I'm sure they're extremely important. The truth is, we didn't do any measurements on that. We actually didn't make any measurements in that regard, but I think they are important. Macrophage, macroglia. We were interested in doing that for a while, and we started. We bought some equipment to try to do some of that work, but there's so much to do, and we just got sidetracked and never got that work off the ground. But it's a great question, and I think it's important.
Fernando Peña, by the chat, he is asking, why dopaminergic cells stopped during GABAergic stimulation when GABA indeed became excitatory after alcohol treatment?
Yeah, when we're actually giving the stimulation, the measurement goes off scale, so we just cut that out. You can't make the measurement while you're stimulating. It just saturates your amplifiers. Usually we'll protect the amplifiers by having a cutoff switch. But if we don't cut it off, it will saturate the amplifier. So you can't measure at the moment that you're stimulating. But as soon as you stop stimulating, you can either see the excitation or the depression. And we followed the whole time course. I just didn't show it all to you, but we followed the full time course of it, being excited and then coming back to normal, or being depressed and coming back to normal.
Very good. Dr. Isabel Mendez wants to speak. Go ahead, please, Isabel.
Hi, thank you very much for your excellent talk. For me, it was very interesting, the effect of corticosteroids, because they are hard immunosuppressors, because they have a circadian rhythm. So my question is if you have tested the effects of nicotine under chronic stress versus acute stress.
Well, we may not have looked at everything you would want us to look at, but we have done a series of experiments which were almost ready to be published, in which we did chronic variable stress during adolescence. That is, this is a pretty severe stress in which the animal is, during adolescence, that 2-week period, is exposed to 7 different stressors that are randomly given to the animal, so it doesn't know what's coming. They include fox urine, being put on a shaker, being put in a cage with a small amount of water, being put on a rocking platform. And this goes on for 2 weeks, off and on randomly. And then those animals show basically the same kinds of problems. They drink more, the circuitry is altered in comparable ways. You know, we don't look at everything, but the things we look at seem to be holding true, very comparable to the acute stress. And that's why when I said adolescent stress or adolescent nicotine, we don't have all that work completed, but we have done it. But we haven't looked at things like immunosuppression or even change in the GI tract, because there's a link to even those factors, but we haven't looked at that.
Thank you very much.
Thank you for the question.
Very good. Dr. Gina Quirarte has 2 questions. The first one, well, she says, congratulations, wonderful seminar.
Thank you.
Have you seen if adrenaline also interacts with nicotine?
We have done experiments on the locus coeruleus, and the locus coeruleus and dopamine we particularly looked in the hippocampus, their influence over memory events. And it's very likely that nicotine does activate that center, but noradrenergic actions are different from those of dopamine, and the dopamine and noradrenergic signaling interacts, and it's somewhat complex. We've published some work on that, and we have another paper that's going to come out. I'd like to say soon, but maybe not. It's going to come out probably this year. I'd say for sure this year. And it's kind of complicated. Sometimes it looks like noradrenergic signaling is mimicking dopamine signals in some way, but in other ways it is not. And sometimes where there's no dopamine signal, the noradrenergic signal behaves differently than when the dopamine is there. So it's not simple, but I think it's very likely nicotine does activate that center. And what it does is not, needs some studying before you can really start drawing conclusions.
Her second question, is there a role for mineralocorticoid receptors if there is an interaction with nicotine receptors?
Yeah, I mean, that's a good question. The mineralocorticoids are just as present as the glucocorticoid receptors, and we coincidentally picked the glucocorticoid receptors for some obvious reasons. We have not gone back to study the mineralocorticoid receptors because this worked so well. Everything just worked. And so we've never really gone back to see what happens when you block the mineralocorticoid receptors. But it's a very good point. It's an area of study. I'm not aware of other people having studied it carefully either, but it's a very educated question.
Maricruz Resendiz says, thank you. Very interesting results. And she has two questions. Did the animals with the two-bottle choice paradigm between water and ethanol had the same exposure to nicotine during adolescence before the treatment with TCB2?
In some cases, yes. In other cases, no. Where you see elevated drinking, the answer is yes. But in other cases, they didn't. These two papers have just come out very recently. One in the Proceedings of the National Academy of Science, and the other, I think, just came out this month in Addiction Biology. In the first set of papers, we did treat the animal. We did pre-treat the animal with nicotine or stress, and they had elevated drinking, and the 5-HT2A receptor brought it down. But I think in other experiments, when we had this chronic drinking, we think that the ethanol itself, when you drink that long, if you notice, when the animal drinks, he escalates his drinking with time. And we think part of this escalation is that ethanol is doing the same thing to this GABAergic circuitry that nicotine did, only it's getting there in a different way. It might not be working through the glucocorticoid receptors. It might be working through a different pathway. So I didn't show you this, and none of this is published, but cocaine does the same thing, and it does not work through glucocorticoid receptors. It also downregulates KCC2. And we believe, and as a matter of fact, we have data unpublished that ethanol does the same thing. It also downregulates KCC2. So the use of the addictive drug itself, by changing its circuitry, leads to its own enhanced drinking or its own enhanced use. So drug use itself leads to a positive feedback through this mechanism, and probably through many others, that enhances drug use with time. So it was very clear in those experiments that I showed you, the escalation of the drinking in the first 4 weeks. And then after we got to 20% ethanol, it still went up to week 4. And only after 4 weeks of drinking did it begin to level off. So we think it's during that period of time that KCC2 is being downregulated, and that's why TCB worked. I should tell you another aspect of those experiments so that you don't go try to do these experiments and find, oh, geez, this didn't work. We selected the animals that drank a lot. So we took this population of, I'll make this up, let's say there were 100 animals, and we picked and we went, what are the 50% that drink the most? These are the 50% that drink the most. Let's go study only them and treat those with TCB. And the reason we did that is the literature is starting to go that way. There's I'm going to mispronounce his name. He's from, I believe he's from Sweden, Hyalure, who he actually analyzes hundreds of animals before he picks the ones he's going to go study for alcohol drinking. And just like in humans, many of us can drink and have no problem, and only a small percentage of us will become alcohol dependent. Well, he selects hundreds of animals, has students working on this all the time, and of the hundreds of animals, he only finds the few percent that seem to act like addicted alcohol drinkers. And then he just goes studies those wild type animals. Well, we took advantage of his point of view, only we don't have as many people to do the work. And we looked at just the 50% of animals that drank the most. And in that case, we saw this big effect of TCB. I hope that answers your question.
Maricruz, second question. Did you evaluate any differences given by sex?
We're doing some of that now. It's in its early phases, and there are sex differences. But for instance, it's known that there are sex differences in the development of the midbrain GABA system, and there are differences in the adult rodent GABAergic system of the midbrain. Given that you know that, you might anticipate that there are going to be differences in drinking, as epidemiology has also pointed out for drinking. So some of that work is underway right now. We only have one student who's working on it. So that's going to be a while coming. If there's something there, I anticipate there will be, but none of the work that's published to this point, or even the unpublished work that I talked about some at the end today, I'm pretty sure that's all males. That the male-female work that's being done now, I don't think has gotten into my slides, the newest work.
Benjamin Villasana asked, may the effect of nicotine on dopamine release leads to anhedonia?
Yeah, I mean, it probably does. I mean, I think that's part of what addiction is. When we had a paper in biological psychiatry a few years ago that showed that after long-term nicotine use, that we did fast scan voltammetry and measured dopamine signals. And what we found is that indeed there was suppressed dopamine signaling in these animals. And part of what the animal's trying to do is get that back. In fact, what we showed is when someone tries to quit smoking, there's a couple-week period of time in which there's suppressed dopamine in the animal. And if you cut slices from those animals, the dopamine signal is suppressed. But if you give that animal nicotine, then the consequences of nicotine has a dramatic release of dopamine. That is, the level that you get the dopamine to is the same as if you gave a control animal nicotine, but the control animal is coming from a baseline here and going up to there when you give nicotine, whereas the other animal is down here, it's much lower, but when you give the nicotine, it goes up to the same height. So in a sense, it's feeling a bigger surge of dopamine during its withdrawal period. So it speaks to why when someone's withdrawing and they have a cigarette and they think, oh, I'll just have this one, why do they so often fail and then go back to smoking cigarettes? Because that one cigarette gives a bigger jolt than a cigarette they might have smoked a year ago when they were smoking normal cigarettes or a control person smoked. It shows that withdrawal, the period of withdrawal, is very vulnerable to relapse. So yes, they do have anhedonia, they do have suppressed dopamine signaling, and that's part of why they relapse.
We have a question from the YouTube transmission. Do you know if the relationship between stress, nicotine, and alcohol consumption is similar in both sexes?
Yeah, that's a very good question. As I said, we're doing some of that work now. I don't anticipate it's going to be the same in both sexes, but we really don't have those data yet. As I said, we only have one graduate student who's working on that. So I expect it's going to go slowly as he progresses. So if anybody wants to do that work, go ahead. You're bound to beat us. You're bound to be faster than we are.
Very good. Let me ask you another question. In your experiments, is there a role for alpha-7 nicotine receptors?
These are good questions. Yeah, I mean, I think it's pretty clear that nicotinic receptors of both kinds are involved in the overall addiction process. The thing that's kind of unique about the alpha-7 receptor is that it has a low affinity for nicotine. A higher affinity for nicotine. So when someone smokes a cigarette, there's a slow rise and there's a peak of nicotine that happens pretty fast, but then a smoker maintains this low level of nicotine throughout the day. That low level of nicotine will tend to desensitize the beta-2 type, the standard type of nicotinic receptor. But the alpha-7 receptor will not be desensitized, but rather it will be slightly activated. It'll flip open off and on now and then. So even though alpha-7 desensitizes very rapidly, it recovers after a time from desensitization. When you get low levels of nicotine, it will continue to flip off and on now and then stochastically from these low levels of nicotine. So how beta-2 type receptors, the standard beta-2, alpha-6, alpha-4, these standard receptors are clearly the main receptor during the addiction process. By far, that's been shown by a number of labs, including our own. But the presynaptic effects of alpha-7 are also important. And even though I bet you could produce addictions without the alpha-7 receptor, if you block them, I bet you could still produce addiction to nicotine. Its overall effect on the learning process, synaptic plasticity, the broad process of addiction, I'm sure they're involved. But I don't want to confuse you. The main receptor is the beta-2 type receptor, and in the midbrain, it contains alpha-6 as well as alpha-4.
Thank you very much. Well, it seems that we arrived to the end of the seminar. No more questions. No more intervention in the chat, and no one wants to speak. Then, John, Dr. Dani, thank you very much.
Thank you. It was really I was flattered to be invited. I wish everybody well. Hope it all goes well. Thank you very much for the invitation. Bye, everybody.